So you know, I'm not. But yeah, now you'll just have. Oh, no. No, go to.
Good man.
I've done a printer and it comes with it. And then one of them.
So.
I'd be more scared of you see my face. So it's much easier. This is gonna. First of all, what? Well, if it's mad. Flying pots of air from there.
Yeah, I was like, what?
It is read on my list of things to do. I know some like I just think the lecture is long, but I guess the updates part is like and then yeah. Well, you know, no, like class notes.
You're bad to go to the bathroom. Show you're I'm like, I walked around the floor. I just, I wouldn't. Oh, yeah. Something like that. I know exactly. Very it's actually like. Oh, I know. You guys is like.
Oh, wait, you're not. How's it going, everybody? Woo! We're Wednesday. I'll tell you, I got the scan trend reports back from your exams. Okay, I miskeyed one of the questions. But I think with like that shifting, I think the class average, and this means I need to write harder exams. Your class average was like a 91/92. I have not graded this. That's not truly the class average yet. Well, we'll see how that kind of happens. Just in full transparency, and maybe this is something you might find helpful to adopt too. I've got so many different things going on in so many different places that this semester I started my to-do list. This is my 443 to-do list, and you'll see I need to post Thursday's lecture material, look over the quiz, and then open, make sure it's correlating to this week's material, grade exams, post lectures, and post study guide. All these things are highlighted in red and Wednesday. So this is what I've been doing, and I found it so helpful because at the end of the day, I just dump my to-do list out of my brain, and I can kind of like move on with my life a little bit, which is a little bit nice. But then I also color code it because I'm super neurotic, and red is like high priority. I think blue is like low priority, and yellow is like, please get to it sooner rather than later. So this is just something I've been doing. I don't know as pharmacy students if you're like having to juggle work, family, and all that other stuff, but I'm just showing you also that you're highlighted on my list. And so all of those things I just showed you will happen today, this afternoon, okay? Maybe I give you guys too much of an insight into my brain. Okay, before we get into lecture specific material about content, does anybody have any questions about what we discussed yesterday? Like I said, yesterday is kind of like the end of big picture introductory material for kind of pharmacology and toxicology. And so now this is our first opportunity to really start to dig into like a specific system, okay? And I really intentionally kick off with the autonomic system because it's advantageous for you because as you'll see with autonomic pharmacology, this is you're gonna see very clear implications for cardiac pharmacology, which we will dig into it deeper come beginning of spring semester. But I find that it's nice to kind of maybe see that material twice in this course. But we'll talk very grossly about autonomic pharmacology, cholinergic agonists, antagonists, so on and so forth. And then we'll kind of transition into neuropharmacology specifically. So I haven't posted this study guide for this week, but if you are if you like to fill out your study guide as we go through lecture, you're probably pretty safe pulling this study guide set of questions up here. I imagine the study guide that I eventually post will not differ greatly from this, right? And some of these questions I've actually worked into the lecture themselves very explicitly. So yeah, we'll just be comparing sympathetic and parasympathetic systems. Again, this is going to feel a little bit like a review from pathophys, but I intentionally build in these reviews because I assume you're learning so many different topics that it's nice to have a bit of a review before we go into it because you probably haven't maybe thought about autonomic pharmacology for a while. We'll talk about neurotransmitters. We'll talk about pre and post ganglionic cells. How do neurotransmitters differ from neuropeptides? And what is the enteric system's role and how is that role kind of mediated and what type of neurotransmitters do you see there? Okay. So again, if we look at kind of overall nervous system organization, we're kind of focused down here for this set of lectures, but for a majority of the rest of the semester will be over here in kind of the central nervous system. So the first branch here is central versus peripheral nervous systems. From the peripheral nervous systems, you have both sensory and motor. And it's almost kind of a misnomer to say the autonomic is a subdivision of motor, because as we'll see, there is sensory input giving information to the autonomic nervous system on how to respond. So we'll talk about that later today. From the motor system, we have somatic versus autonomic. And then when I say somatic, that's your musculature, and the autonomic would be the kind of automatic nervous system that we'll be further discussing that has divisions in both sympathetic versus parasympathetic. Okay, so first set of questions I have for you, somatic versus autonomic. How do they differ from one another? How are they the same? And why does this kind of matter to you as a pharmacist? So I'm posing these questions again, just to kind of keep you engaged and chatting with your group. So I'm just going to give you a few minutes to discuss within your group these three questions. So somatic versus autonomic, if you were going to describe how they're different from one another, what would you say? Are there any ways in which these two nervous systems are similar? Okay. And then if they are similar or dissimilar, again, why do you care kind of as a pharmacist? So go ahead and discuss for a few minutes.
Yeah, between those two and big motors, those things are most feedback. Yeah. So, but
I think like centuries gonna be more accurate versus accurate, which is we'll talk about.
Yes.
yeah, we don't target. Yeah. Never heard. So no one
In a good size, but yeah, no.
Yeah. Oh, we already went over. Yeah.
Okay. Does anyone want to share out? So somatic versus autonomic, how do these two systems kind of differ from one another?
They get different signals from different parts of the body, like the autonomic system has like the ganglia.
Yeah, so their structural organization is different. Okay. The areas in which they maybe send information to might be slightly different as well. What about Yeah, so this kind of idea of conscious control, right? Autonomic nervous system, for the most part, we do not have conscious control over, with the exception of there's some types of physiological systems that we can consciously control with the added input of the somatic nervous system. So the example that I'm specifically thinking of is respiration, right? Like you respirate kind of whether you want to or not most of the time, right? But you can use your somatic muscles to kind of override that and hold your breath or slow it down and all of those fun things. One of the reasons I think that maybe meditation is so effective is because it does engage that autonomic part of your body too, right? And so they differ from those levels. Again, autonomic nervous system won't be under conscious control, and somatic nervous system generally is for the most part. How are they the same? Are there any similarities within these two systems? Dr. Peacock, I heard you say something. They're both divisions of the nervous system for sure. Yeah. Anything else? I mean, they're both interacting with their efferent projections with some type of musculature sometimes, either smooth or somatic. Neurotransmitter wise, both are going to use acetylcholine. Okay, so we'll talk about that a little bit later on, but sometimes it's nice again to think of these similarities versus differences. And so why does this matter to you as a pharmacist?
There's drug targets.
Yeah, lots of drug targets in this system for sure. We'll talk about, you know, the adrenergic, the subtypes, as well as the cholinergic types of receptors that we target pharmacologically. But again, keep in mind when I just was pointing out the similarities here, if you alter acetylcholine, you might have both somatic and autonomic effects. Okay, so that's one of the reasons I made you try to think about the similarities between them. Okay, so this is, I mean, you could easily turn this into a table. I just kind of like this kind of side by side comparison between the two. Again, this is most likely a review, but it's nice to kind of just see this information fresh. Where your autonomic nervous system, again, being biology, has several different names. The visceral system, the vegetative system, the involuntary system, mediates functions that are not under conscious control, as we just discussed a second ago, and kind of has these structures in the peripheral body that are nerves, ganglia, and plexus that interface with the heart, blood vessels, glands, and other visceral organs. And we'll talk more about that structure moving forward. And then distal synaptic junctions in the autonomic reflex arc occur in the ganglia that are entirely outside of kind of the cerebrospinal axis. That's also part of our autonomic responses. Keep in mind, you can see here postganglionic autonomic nerve fibers are generally non-myelinated. And why does that impact you, or why do you care? That non-myelination is going to kind of slow down the rate of conduction in those instances. Somatic nervous system, pretty straightforward in the context that we're talking about. Again, that's not our primary focus for today, but again, it's innervating that skeletal tissue. Networks are kind of absent from the somatic system. And then those motor nerves to skeletal muscles are myelinated because, again, just step back evolutionarily, right? When you're trying to move, you want that business to happen as quickly as possible, right? But the two are very much interrelated. Keep in mind when we talk about kind of that fight or flight response, you are going to see increase, you know, vascular pushing of blood flow to the somatic system for that kind to mediate that fight or flight response. So the two are related in that way too. Okay, so I like this particular figure because it does a nice job of kind of comparing and contrasting the synaptic differences between the somatic nervous system, right? So this first column here, you have the breakdown of the somatic nervous system and our autonomic nervous system. Here are the neurons that are a part of the central nervous system. And in most instances, these central nervous system neurons are going to be found in the spinal column, with the exception of some of our autonomic neurons are also going to be part of like our brainstem and our cranial nerve system, especially when we talk about the autonomic system. So here we can see for somatic, pretty straightforward. Here you have the projection out, the release of acetylcholine, and then again, the effector organ is going to be skeletal muscle. Now, I'm going to harp about the neurotransmitters that mediate this process a lot, because those neurotransmitters are going back to kind of how we've laid out our foundation. Those neurotransmitters are considered our endogenous ligands, right? We've been using that term a lot. These neurotransmitters are going to be our endogenous ligands that are going to be binding to our final tissue targets. And that's where we can start to introduce those pharmacological interventions. So knowing the receptor subtypes that are at those target tissues, that's what you're targeting as a pharmacist, right? That's what you're trying to agonize, antagonize, partially agonize, all of those things is at the level of those target tissues. Keep in mind though, in the case of, let's say, the sympathetic division of the autonomic nervous system, you have kind of this intermediary step here where you either have ganglia or the adrenal medulla that are going to also have points of regulation. In this case of the ganglia, they're going to release norepinephrine, and those are going to hit things like smooth muscle. In the case of the innervation of the adrenal medulla, the neurotransmitter here is again acetylcholine, and that's going to lead to the release of norepinephrine and epinephrine into the vascular system. In terms of the parasympathetic division, we have a release again of acetylcholine, and again, we'll see acetylcholine released here, and that can again affect either smooth muscle glands or cardiac tissue. Which neurotransmitter fits with what system? Hate to say it, but it's important, right? You do see kind of repetitive uses of acetylcholine. Keep in mind, acetylcholine is kind of at the preganglionic cell release here, and is at the kind of pre and post ganglionic cell in the parasympathetic division. Okay. Yes. Yeah, so anywhere where you see kind of like these bubbles, that's representation, that's the myelin being represented. In the case of kind of this post-ganglionic cell, you can see kind of the dotted line, that's not myelinated. And then in the case of the adrenal medulla, it's just releasing those neurotransmitters directly into the vasculature, which isn't necessarily like a neurotransmission event. It's not necessarily a neurotransmitter, or it's not necessarily a neuron that's releasing those neurotransmitters. It's just going to be from the adrenal medulla itself. But good question. Yay. Any other questions? Okay. Okay. So as we talk about kind of the autonomic nervous system, sometimes it's nice to kind of go back up into the central nervous system and see kind of like what are the larger structures that mediate some of these automatic or autonomic responses. And again, in these higher structure areas is where you see the interaction of both the somatic and autonomic nervous systems, right? The example that I just gave a second ago is if you undergo a fight or flight response, like if I said, everyone pull out a piece of paper, we're going to do a pop quiz right now, right? Your blood pressure might go up a little bit, yeah, right? You might have a little bit of a fight or flight response in you, and your vasculature would start shuttling your blood to your musculature to actually evade the physical threat that your mind is telling you to interpret, right? Now, autonomic reflexes remain intact again at the level of the brainstem. So if you have an individual that's actually transected, meaning kind of this area is cut, you can still see sweating, blood pressure alterations, vasomotor responses, temperature changes. Again, you're still able to exhibit autonomic nervous system's response in the absence of higher order cognitive thought. Okay, so I didn't really explain this experiment fully before I started going through it. If you literally cut off the brainstem from the cerebrum, again, the brainstem is literally the stem of the brain, you can still have conserved autonomic responses based off of the structures that you see here. So as long as the thalamus, midbrain, pons, and medulla oblongata are all kind of intact, you'll still see autonomic nervous system response. Keep in mind, there are areas within this brainstem that are important, not just for autonomic response systems, but even levels of consciousness, right? If you sever specific areas of the brainstem, that's when you're going to have kind of brain death occur, and they'll have lack of consciousness that happens. But integration of both somatic and autonomic again occur at these structures of the medulla oblongata, the hypothalamus, which is also going to get activated and lead to release of some hormones, and then the subthalamic nucleus, which is also just kind of a subregion of the thalamus itself, which is a major relay station for all sensory information except olfaction. Yeah. Okay, so let's step back. We've been talking about sympathetic versus parasympathetic, but again, I'm going to ask you this very kind of same set of questions. Parasympathetic versus sympathetic. First,
If you don't clearly have the rest and digest and fight or flight correlated to those two terms, now is a good time to get that really solid in your mind. So that's maybe the first question you should talk about as a group. How are they different? How are they the same? And then again, why does this matter to you as pharmacists? You could probably give me the same answer as you gave me last time, is like, it's a drug target. Totally true. So again, take a second and chat with your group as to, again, how they're different, how are the same, and then again, why does this matter?
Yeah, I think we got tissues. Yeah, they don't.
Oh, so that's exciting. I guess.
No, I don't know. Like the venue is kind of in like the middle of forest.
Okay, I think I'm hearing some conversations devolve into other subjects, which means it's time to kind of come back together. Okay. So sympathetic versus parasympathetic, how are they different? One ups our system, one's kind of down our system. So one activates that kind of response to a threat, the other one calms us down, and I would say kind of keeps us in homeostasis most of the time. I kind of want to get on my soapbox here for a second, right? We're meant to be in this homeostasis state more often than not, but I feel like honestly the way we live more often than not is in that activated fight or flight response. I know I feel that way. So again, going back to like evolution and kind of the environment that we live in, and it kind of has some interesting societal implications. But that's just me on my soapbox for a second. It's just kind of like, how can we again get that homeostasis back in check? How are they similar? How are they the same? What? They're both autonomic, and they both at some point in their signaling do use acetylcholine, right? That's one way in which they're similar. Why does this matter to you as a pharmacist? I kind of keep giving you this answer that specifically related to the neurotransmitter acetylcholine, right? If you give a nonspecific cholinergic agonist, it could affect both systems. So just, again, I'm just trying to get you to think about that and why this is interesting and why we have to learn the specifics of this at all. Okay, so if you haven't figured this out yet, 9 times out of 10, when I ask you a question, the next slide has the answer. Which I'm guessing many of you picked up on by now. But here we have the sympathetic versus parasympathetic systems. Here we have the sympathetic system, which is our fight or flight, and our parasympathetic system, which is our rest and digest. Again, our sympathetic nervous system should just happen when we have these like challenges to our system, and that will dilate your pupils, inhibit salivation, increase your heartbeat, again, increasing your oxygenated blood getting to your somatic muscles, relaxing your airway so you can breathe and again, take in more oxygen, inhibit stomach activity, because if you are running away from a bear, you don't need to be digesting your lunch, right? All of your blood flow needs to be going to your musculature and evading this threat. Inhibit the gallbladder, inhibit activity of intestines, secreting epinephrine and norepinephrine, and then relaxing the bladder. Hopefully not too much. But again, that is kind of why that can happen in moments of threat, is because of the mediation of the system. Rest and digest, like I said, is kind of hopefully where we're living more often than not with constricted pupils, salivation being stimulated, slow heartbeat, constricted airways, a little, to a certain extent, activity of stomach, gallbladder, intestines, and contraction of the bladder. So hopefully you're just kind of hanging out and chilling most of the time, just worrying about digesting your food. Okay. To oversimplify it. Again, if you can't correctly kind of match, parasympathetic with rest and digest and sympathetic with fight or flight, strongly suggest you get into that place sooner rather than later, because every subsequent kind of decision that you make about this regarding the pharmacology is going to kind of be determined whether you're trying to activate or slow down that system. So that's why we're even kind of spending time here. Okay, in terms of neurotransmitter differences, this figure we just looked at, so we got some information about, again, the preganglionic cell releasing acetylcholine, and then later releasing norepinephrine in our sympathetic division. And again, a sympathetic fight or flight or rest and digest. Fight or flight. Yep, we're ramping up the system. And that's, again, if you kind of want to work backwards from maybe information that you already know, norepinephrine and epinephrine are going to bind to those adrenergic receptors, which again is where beta blockers also act, right? So that hopefully conceptually makes sense in your mind. And then our parasympathetic system has acetylcholine being released at both of those. This figure shows you the exact same information. It's a little less decorative, doesn't show anything about myelination, but sometimes I like to give you a couple different options in how you look at the material or the figures, because again, looking at it this way just might be a little bit clearer. But it does just indicate, again, the parasympathetic system that we're looking at down here, acetylcholine and acetylcholine is being released in the parasympathetic, and then the sympathetic, again, acetylcholine, norepinephrine. And in some synapses for sweat glands, you actually do see acetylcholine being released at both synapses. And then again, that sympathetic output to the adrenal medulla leads to the release of epinephrine. Okay, again, this is something that I know we discussed a little bit last semester, but knowing the neurotransmitters and which synapses they are being released at, super important, again, to our pharmacological interventions that we are later going to use. Okay, so I'm just trying to point to these points that if this does not feel comfortable for you, I don't know, I kind of feel like I might need, would you guys like a worksheet related to this? Let's, okay, like want to exit out and put it on my to-do list right now. But yes, worksheet related to this might be kind of nice just to really pull out like sympathetic versus parasympathetic, and then the neurotransmitters responsible. Okay, now we have divisions of the peripheral autonomic system. I know this particular figure might look a little busy. We have, again, our sympathetic or thoracolumbar outflow, and our parasympathetic or craniosacral outflow. Now, why this is it may be helpful is when we're looking at these two divisions and kind of the outputs from them, keep in mind your parasympathetic might have a higher relationship to kind of the brainstem area and your cranial nerves. So we'll talk about that a little bit later. Okay, so as we are talking about the nervous system, again, very big picture, 30,000-foot view of the nervous system. The function of the nervous system is to take in information and produce a response. We can all agree upon that. Now, the autonomic nervous system, same situation, where the information that you're taking in from what we call the afferent division, towards that information goes towards, your dorsal root of the spinal column, and then goes up to your brain, kind of goes up to the brain, that sensation goes up to the brain, gets processed, and creates an efferent response. And in this case, we see the autonomic ganglia going out to, again, the preganglionic fiber to the postganglionic neuron, and this is the autonomic efferent division response. If you're not comfortable with these terms, I always think of efferent as being the exit, and then by process of elimination, I can remember what afferent means. So if this, again, nomenclature is not super familiar to you, you can use that trick or whatever trick works for you. But I just want to make sure we feel pretty comfortable with the afferent versus efferent information that's being processed. Okay, so first we are going to talk about what type of information goes into the autonomic nervous system. So these are the visceral afferent fibers, meaning that you have neurons that project to your stomach, right? That tell you when you're hungry. We, again, can all agree upon that. that would be an afferent projection system that projects into your autonomic nervous system and causes a response like the stimulation of salivation. So we have two sensory systems that transmit information on the status of the visceral organs. First, you have the spinal, which is considered the sympathetic visceral afferent system, and the cranial nerve or parasympathetic visceral sensory system. Now, I've color-coded sympathetic and parasympathetic because it's color-coded on this particular image as well. So you can see kind of the blue up there for cranial nerve 10 and cranial nerve 9. That's information that's going to the brainstem through those cranial nerves. So that's the afferent information going through the parasympathetic system, okay? Why would you have afferent information going through your internal carotid? What information is that telling your body? Blood pressure, yes. We have baroreceptors that sense pressure in our internal carotid. That's giving us information about where our blood pressure currently is at. Over the summer, I discontinued my blood pressure medication and I started to have some pain in my neck. And my doctor was like, why are you talking to me about your carotid? I was like, because I know too much. But I thought, I was like, oh my God. I started freaking out. I was like, I'm going to have a stroke if I don't go to the doctor. So I went to the doctor in August. Anyway, if we look at the spinal system, again, this is a sympathetic system. You can see there are a lot more inputs from those areas that you're going to receive information from. Again, the question here is like, how much of this should you be committing to memory and really knowing? I think, again, the big picture of that you have a lot more afferent inputs from your sympathetic system, the spinal visceral afferent system, than your parasympathetic system, probably is worth noting here. Now, the spinal sympathetic system that you see here on the left side of this figure, again, we can even further divide that into kind of this visceral afferent system that conveys sensation related to temperature, tissue injury of mechanical, chemical, or thermal origin, and these collaterals that converge with the cranial visceral sensory pathways virtually kind of at every level. So you have lots of levels of integration that are occurring here, especially on kind of, again, the sympathetic side. If we go over to the blue kind of parasympathetic side, again, the cranial nerve inputs that we see kind of there at the top. I guess I could do this too, here at the top. You're getting mechanoreceptor and chemoreceptor information. Again, mechanoreceptor is a type of receptor that opens based off of mechanical deformation, so things like pressure and those baroreceptors that I was just talking about. And then also chemoreceptor information as well, especially within the arch of the aorta. They enter through 4 different cranial nerves: the trigeminal, the facial, the glossopharyngeal, which is cranial nerve 9, and the vagus is 10. Okay. I won't necessarily, I'm most likely with the cranial nerves will give you the name and not the Roman numeral. Before, I've actually had students who were like, I never learned Roman numerals. I was like, okay, well, I'll just use the name then. So we'll just go with the names versus the Roman numerals with those. Or I might just include both of those pieces of information so you don't need to memorize the cranial nerves on top of everything else. They're transmitting this visceral information to the internal face of the head and neck, tongue. Again, you also see some pelvic viscera that are innervated by nerves from the 2nd through 4th sacral spinal segments, as well, as you can see shown here. Later, we'll talk about the sympathetic chain ganglion as part of this organization. Okay, I've been yapping at you for a while. As we're talking about the neurotransmitters like acetylcholine and norepinephrine and epinephrine that are being released, in addition, the neurotransmitters at the sensory fibers, for the afferent sensory fibers, we have substance P, CGRP for our sensory neurons, SST, VIP, CCK, ATP, as well as some additional neuropeptides in the dorsal spinal cord as well. So I just wanted to see what y'all knew. Go ahead and take out your phone. Before you do, though, I would like you to pair up with somebody. Pair up with at least one person on your table. As you pair up with them, sign the back of your attendance cards as well. And then have one of you pick up your phones and provide your answers for this question. And then.
Yeah.
Has anybody put any answers in here? Oh, okay, cool. No. I just wanted to make sure I wasn't having a tech issue. Okay, cool. I just want to make sure I didn't make a prompt. Okay, good. I've got a response. Thank you. Was that you, Paige? I like the trail at the end. I was like, looks like you fell on your keyboard. Tommy, it is the same ATP. It's not ATP? I said it is ATP. It is ATP. It's the ATP that you know and love. Yeah. But I don't know. That is not the VIP I'm talking about. The type of, can be one of the precursor neuropeptides or opiate signaling. Heparin, endorphins. I went by to the different opioid receptor symptoms. Well, enkephalins and endorphins are actually different molecules, but they have similar targets, if that makes sense. They just have different affinities for those targets. Yeah. When would you, enkephalins and endorphins, I think really sense is probably similar kinds of stress or pain or high. Yeah. Okay, you guys ready to see your answers? Yes. Oh, I'm sorry. Our folks at home are like, I can't see anything. Okay, here we go. So here we have some of our abbreviations here. ATP is again adenosine triphosphate. TCK, cholecystokinin, somatostatin. Yeah. Okay. Very important person. Yeah, okay, so many of you came up with, some answers for sure. Okay, here we go. Here's our answer sheet, kind of. Again, calcitonin gene regulating peptide. Sensory neurons are going to be nerves that detect and transmit sensory information. That wasn't one of them. Somatostatin, vasoactive intestinal peptide. Cytocholecystokinin, and then ATP was adenosine triphosphate. Again, in this context, we know ATP to do a lot of different things like being an energy carrying molecule, but in the purinergic signaling can also be a endogenous ligand as well. Yes. I can post it. Yeah. Yeah, no problem. Maybe I'll make it part of our worksheet too. But yes. So the last slide, okay. So when we first started talking about autonomic nervous system signaling, I kind of pulled out, we have acetylcholine, we have norepinephrine, we have epinephrine. These are our neurotransmitters that are going to be signaling in the system. And then this slide is actually showing neuropeptides. Again, this is something I feel like I've harped on a ton in almost all of my classes, is like, how do neuropeptides differ from neurotransmitters? And again, they have a lot of different properties. So the first part of this question, I kind of like to ask yourself, how do they structurally differ from one another? So again, chat with your table. If you did not if you didn't do your the last question, just make sure that happens before you leave today. But go again, have a quick discussion. How are neurotransmitters and neuropeptides different?
One whispers. That's what it sounds like. Yeah. Well,
Yeah.
Sorry, something in my throat. I promise. Okay, really quickly, let's keep moving here. Neuropeptides versus neurotransmitters. Structurally, how are they different? Peptides are bigger. Maybe even a better question is, where do they come from? Neuropeptides kind of look like the peptide word that we know associated with proteins. And so neuropeptides are actually proteins. They're encoded from our genetic material. It's kind of wild, right? They're encoded, they have their own gene, they have their own mRNA, they have their own peptide product. In a lot of ways, as we'll talk about later when we talk about the opiate system, neuropeptides are almost like prodrugs in the fact that they have to be cleaved in very specific ways to yield their final signaling molecules. So they're pretty large in comparison to neurotransmitters. When we look at the structure of neurotransmitters, we're going to see that they're probably just modified amino acids. We're not going to get crazy into the med chem. You've got a whole nother class to torture you in that way. But I'm guessing in that class, you will also talk about some of the modifications to those amino acids to yield neurotransmitters. Okay. So here's a nice table kind of comparing and contrasting. As I said before, if I ever ask you a question in class, usually it's on the next slide. Sometimes I hide them from you though. You can see the neurotransmitter is synthesized in the synaptic nerve. Again, because a neuropeptide comes from the central dogma, it is synthesized in the neuronal cell body. Here you see it stored in small synaptic vesicles. Because this is a larger signaling molecule, it has a large, dense core vesicles, literally just physically larger. You have synaptic release against spatial specificity, meaning that one neuron releases this neurotransmitter, and generally, we'll say generally here, it's going to diffuse across and hit the postsynaptic cell. In the case of neuropeptides, because those molecules are so large and stable, they can get released and signal to far-reaching areas. So when we talk about neuropeptides, I know we just discussed things like somatostatin and such, but hormones also kind of fall under the category of being neuropeptides as well. And again, think about your hormones are synthesized and released from your pituitary gland, but affect all areas of your physiology. Neurotransmitters mostly bind to ionotropic, but also to metabotropic receptors. Neuropeptides mostly bind to metabotropic receptors, and you can see in there mostly is kind of a very broad term. Neurotransmitters, because of their size, have a shorter half-life, and neuropeptides tend to be much longer half-life. And again, in a very just general description of them, neurotransmitters are kind of like whispered secrets, very specific. It's like if you have a piece of gossip you want to tell somebody, you're only telling that person and you're whispering in their ear, right? You've got a single one-to-one translation. Versus if you yell out in the middle of class, I love dogs. That's a public announcement, right? Everybody knows that. And that's how neuropeptides work. They hit kind of non-specifically everything. Okay, so as we are talking about neuropeptides versus neurotransmitter, that's kind of a quick aside about some of the afferent sensory neuron signaling molecules. So now we're going to kind of quickly shift over to not just the afferent going to the brain information, but how are these systems organized in their efferent or exiting kind of projections to the physiological areas. So again, to kind of just clearly lay this out, we have our sympathetic nervous system that is fight or flight, which is shown here kind of in a purplish red. We've got our, actually, no, this isn't coded right. So the parasympathetic system is shown on this side of this figure. Our sympathetic nervous system is shown here on the right side of this. Again, fight or flight versus rest and digest. With our parasympathetic system, rest and digest, we see our outputs coming predominantly from those cranial nerves located in the upper part of the brain or the brainstem that you can see here. And our sympathetic nervous system, we can see again, we have this entire what we call sympathetic chain ganglion, kind of like the stopover points before they head to their final points of innervation. In addition to our parasympathetic and sympathetic nervous system, we also have the intrinsic enteric nervous system, which is going to be kind of our gut. You guys have probably heard the term kind of gut brain. Our intestinal system has kind of an independent set of neurons that kind of encapsulate them all, which makes sense if you think about how the peristalsis of the gut works, right? It has to be kind of a contraction cycle that occurs in concert with muscles. And so with that, you've got this interconnection of neurons that line the bowel and the gut to kind of allow that peristalsis to happen. There are as many neurons in the gut as there are on the spinal cord. So again, that's one of the reasons we talk about the gut being its own kind of system and in terms of just even numbers. And then we have the neurohumoral component of the ANS, which is kind of usually considered part of the endocrine system. And again, these are related to hormones that regulate energy metabolism and blood volume. Questions? Okay, we're getting close here. So first we have, again, the sympathetic breakdown of the outward projections or efferent outputs into this area. As I mentioned a second ago, with the sympathetic nervous system, we also have this adjacent set of neuronal clusters called the sympathetic chain ganglia. Okay, now we obviously have neurons that exist within our spinal cord, but what's interesting about the sympathetic chain ganglion is they run just adjacent to the spinal cord and are kind of like the stopover area for this kind of preganglionic and postganglionic cell. Okay, so these are the cells that give rise to, again, the preganglionic fibers and the intermedial columns of the spinal cord. T1, you see it run all the way down to kind of S5. If we're going to break these down into the areas that they're a component of, you have your paravertebral, which are going to be the 22 pairs of sympathetic ganglia that form kind of the lateral chains, the prevertebral that are located in the abdomen of the pelvis near the ventral surface, and then the terminal, again, in few in number, lie near the organs that they kind of innervate in terms of kind of their structure. This is showing you the left chain ganglion, the right chain ganglion. These are kind of going to be mirrored on both sides of your body. You can see how they're even subdivided into kind of your superior cervical ganglion that innervate most of this area is kind of the upper part of your body, your eye, your lacrimal gland, your mucous membrane of the nose and palate, your sublingual gland, your mucous membrane of the mouth and the parotid gland. You can see here, we have this next projection to the heart, larynx, and trachea. Our celiac ganglia that we see here are innervating your stomach, abdominal, liver, pancreas, and adrenal gland, as well as your small intestine. Again, I'm giving you a lot of nomenclature, but keep in mind, the way that I kind of like to think about this is how they're clustered and how those physiological systems that they innervate and how they're clustered is usually paired to what they're doing, right? This is all kind of head and neck stuff. This is kind of more cardiac stuff. This is kind of more digestive. And again, down here, we see kind of large intestine as well, kind of being part of that inferior mesenteric ganglion as well. How are your brains? Full. You're like, you just threw multisyllabic words at us the last like 3 minutes. So I think I'm just gonna pause there. I do have quite a few more slides in this. And so we'll finish up this slide deck tomorrow. And knowing how far we got today is going to determine like how big that slide deck is tomorrow. I'll work on a worksheet to help you kind of organize this in your mind, kind of how I think about it and clearly kind of lay it out. And I'll also build in tomorrow's lecture to that as well. I'll post the study guide, I'll post the lectures. If there's anything else that you think you might need from me, feel free to reach out, send me an email. Otherwise, does anybody have any questions before I let you go today? Okay. Leave your name cards on your table. I will collect them. Otherwise, have a wonderful Wednesday, and I will see you tomorrow.